Today we want to talk about integument or skin. We want to talk about the two tissue components, the epidermis and the dermis, the different layers of the epidermis and dermis, the cells that are involved, the granules that are involved, and we end with some disease states. Thank you. Integument. We want to describe the different layers of the epidermis, describe organization of the layers of the dermis, then describe the structures of the sebaceous gland, eccrine sweat gland, apical sweat gland, and to recognize granules, melanin granules being one of those in skin. Now, skin has a binary origin. The epidermis comes from the ectoderm, from the outside, but the dermis comes from the mesoderm. So remember that organs, which skin is composed of at least two types of tissue, and one is epithelium and one is connective tissue in the dermis. Now, what's the function of skin? It protects us against injury and desiccation. We live in a dry environment. If you ever left a piece of bread out on the table at night, the next morning it'd be very dry. Excretes various substances, thermal regulation. It receives stimuli, temperature, pressure, and pain. It's the basis of recognition, and it gives clues. You can look at a person and tell how old they are. You can look at the skin of a person and tell how nutritious they have been. And then for fat metabolism, subcutaneous layers. So we will concentrate on thick skin and thin skin. The thick skin allows us to see the different layers of the epidermis better, and we will talk more about that. Thick skin is more kind of rough, as thin skin is smooth. And in fact, if we look at a regional variation in skin, this is thin skin is a sole of a foot, for example, and your thin skin may be eyelid, a very thin portion of eye. Sometimes the skin is transparent through the cornea, covers the eye. That's a skin component. And then there's various appendages. You got nails, you got hair, different structures, glands, that is part of the regional variation in skin. Now, the thickness of skin, whether thick skin or thin skin, really depends upon the epidermis. You can have a very thick dermis and still be considered thin skin. For example, the skin around your waist has a very thick subcutaneous layer and hypodermis, and nevertheless it is considered thin skin compared to thick skin. And so we look at the layers. This is thin skin at the top. We have the epidermis, the dermis, and the subcutaneous layer of skin in through here where the fat cells are located. Sometimes the hair follicles go all the way down to the subcutaneous layer, or the hypodermis, as someone might call. And then thick skin, you can see how thick the epidermis is, and then the dermis, and then the subcutaneous layer. So the thick skin would be where you have abrasion occurring, like on your hand, heel, and thin skin would be pretty much the rest of the body. If we look at this thick skin, we can see the different layers: the epidermis, the dermis, and the hypodermis, or the subcutaneous layer, which contains the fat layers. So basically we got the epidermis, dermis, and the hypodermis. Now, if we look at the dermis itself, you can see that has different components too. The bulk of the dermis is called the reticular layer. However, some parts of the dermis interacts with the epidermis, as you see through here, and it's called the papillary layer. Papillary versus reticular layer. And the papillary layer, you can see the dermal papillae project up. So this is dermis, and coming down into the dermis is the epidermal pegs, or they may be called reedy pegs. So this is epidermis coming down into the dermis. These are pegs versus dermal papillae. And you can see these if you electron micrographs, scanning electron micrographs showing you the dermal papillae projecting up where the dermis would be. Also, the dermis provides the nutrients, blood vessels and all, and here you can see the blood vessels which are inside each of these dermal papillae that you see. Now, we can see again that not only do we have dermal papillae projecting up here, but also in connective tissue you have nerve endings, and here is a Meissner's corpuscles in a dermal papillae. The Meissner's corpuscle is a mechanoreceptor nerve ending sensitive to the light touch. So it's kind of light touch. If you tickle yourself, that would be the one that you would feel. You would have more of these in your fingertips than you would the rest of part of the body. The skin, we can see skin here from a monkey. You can see the hypodermis. All these are fat cells in through there. But this is the reticular layer of the dermis, and then the papillary layer of the dermis, and you can see dermal papillae and epidermal pegs of epithelium projecting down into the dermis. Also located in here are the proscenium corpuscles, which look like onions, but you can see several of those, and we'll talk about that later on. Also, you can see sweat glands. This is a sweat gland here, and this is a duct of a sweat gland. So that is the acrosweat gland, the one that's associated with cooling. And here we can see the chicken wire looking stuff, which is the adipocytes. These are the fat cells in the hypodermis. Now, here we can see the obesity is characterized by a thickened subcutaneous layer or hypodermis. And you can see this is an individual from Body Worlds that weighs 300 pounds versus one that weighs 120 pounds. And you can see the hypodermis is very, very thick on this individual. So there's different layers of skin, and we can see those in the palms or soles of your feet. And the top layer, the outer layer, is a stratum corneum, and it's keratinized, flattened, denucleated dead cells. So these are dead cells that are the most differentiated ones that ultimately will be sloughed off. The next layer down is granules that you can see, stratum granulosum, and that's the keratin hyaline granules. And the keratin hyaline granules are mostly the intermediate filaments, which is what persists in the digested cells. And then you have the stratum spinosum, and that is this area right in through there, and you can see little spines, or they're really desmosomal connections, tonofilaments, or the intermediate filaments that attach to desmosomes. And so you can see those there, and we can see those in a minute. And there's a basal layer. So the stratum germinativum or stratum basal right in here, this is a layer that touches the dermis. And this is the mitotic active layer of the epidermis. And so this is a source of cells that differentiate and ultimately die and slough off at the surface. So these are undifferentiated, these are differentiated versions of the same cells. This is the keratinocytes that are in there. Here we can see a sweat gland. There's a sweat gland duct coming up through there, and that sweat duct continues all the way up into here and discharge its sweat on the surface. So we have different layers. There's also another layer right in here, stratum lucidum. It would be the fifth layer that we see. And we can see those here on the skin. You can see how thick this stratum corneum is, stratum lucidum, stratum granulosum with the keratin hyaline granules. We see them over here too. Stratum spinosum, and if you look in here, you can see where kind of the cells are pulled apart a little bit, and they're held by desmosomes. And then the stratum basal, this layer right here, which actually touches the dermis, and it has hemodesmosomes. Hemodesmosomes, half a desmosome, also has desmosomes, and desmosomes is what holds these cells together. And then here we see the keratin hyaline granules. Now, in the stratum granulosum, that we see right through there. We have flattened cells undergoing terminal differentiation process of keratinization, and they form the skin barrier with water, prevents dehydration, one of the functions of these, if they're sealed by the membrane coating granule contents. So we'll see a little later on how those different granules of membrane coating granule contents seal these cells and prevent dehydration from occurring. So we can see the epidermis of thin skin is subject to pressures and friction, and so it needs a desmosome to hold the cells together. And here you can see these are actually desmosome connections, the connections between adjacent cells. We can see those here and here better. What happens is when the tissue is processed, the cells shrink. And as they shrink, the desmosomes still hold tight. And in fact, this is in skin, stratum spinosum, that they discovered desmosomes, because you can see the desmosomes where they are attached, where the cells are shrunken apart from the rest of from the other cells, and as a consequence, you can see the desmosomes which are holding tight between adjacent cells. So stratum corneum, the stratum lucidum up there, stratum granulosum. You can see the granules in through there. Stratum spinosum, where you have the desmosomal connections, and the stratum basale, which is a mitotic active portion. So all these cells are differentiating, divide here, differentiating, and ultimately sloughed off at the top. So the stratum basale has these hemodesmosomes, as we mentioned before, and it interacts, has a basal lamina in through there, and a basal membrane that we see at light microscopic level. This is collagen fibers that we see where it's interacting, attached to the collagen below. Above the base, we have the stratum spinosum, and you can see these desmosomes between adjacent ones. Here we can see where the tissue is somewhat shrank a little bit, and you can see the desmosomes are still holding tight on these, and that's where desmosomes were discovered, was in skin. And here we can see an electron micrograph, the basal layer, stratum spinosum. You can see the spines in through there, which are really the desmosomal connections between these two. Keratin hyaline granules make the stratum granulosum, and then above that you've got the stratum corneum. But you still have cells. These are still the cells, but all their contents have been digested away. You just have the keratin hyaline granules, which provides the content, undigested components of the intermediate filaments, which is really what keratin hyaline granules are. And here you can see the filamentous, but you see the desmosomes are still holding tight of the cell. What's left of the cell, all the nuclei and organelles have been digested. Just the keratin hyaline granules persist in through there. Here you can see where stratum corneum, the cells are all stacked up. Again, they're held tight by desmosome connections, and all the contents of the cells have deteriorated at that time. Cells of the epidermis, you have stratified squamous cells that include various types of cells. You have the keratinocytes. That's the main cell type, and it's from the ectoderm, as we mentioned before. There's melanocytes. Those are the ones that are responsible for pigmentation of the keratinocytes, and these cells are from the neurocrest. There's Langerhans cells, also in the epidermis, and they have an immunological role that we'll talk about in a minute. And there's Merker cells, those associated with nerve endings, and here we see one of those cells. They do make desmosomal connections with the keratinocytes, so we can't separate them from the keratinocytes, but there's a nerve ending that's associated with that helps detection of something in skin. Now, melanocyte is responsible for the pigmentation, and that's from the neurocrest. Here we've got the neurotube, neurocrest. Those cells migrate out. They migrate down here into the basal of the epidermis. They also can be found in the dermis, and they're in the hair bulb. So right here, which is a source of mitotic activity for the development of the hair, you have melanocytes which give the pigmentation to the hair. So they come from the neural crest, those cells that migrate in through. And here we can see them. These are melanocytes. It's a dendritic cell that has processes that go from itself to other cells, and you can see those here. They do not make desmosomal connections with adjacent cells. As a consequence, they appear as a clear cell that on the stratum basal. So they're located on the base. And here you can see those. If you took the epidermis and looked from the view that the dermis has of the epidermis, you would see these dendritic cells, these melanocytes located in through there. And as I mentioned, they are clear cells. You see one here, see one there. It doesn't make desmosomal connections, and so whenever it contracts during the processing, some of the cytoplasm pulled away, and it looks like a clear cell, as we see there in the basal. Now here you can see tyrosine is involved. You've got raphe reticulum, Golgi, to produce the melanin granules that then the melanocyte produces the granules, and here we see the melanocytes with projections therein, and they pass that on by cytokine secretion to the keratinocytes. So here you can see there's melanocyte budding off the melanin granules with a membrane, and is going to a specific cell. So then it will be taken up by the keratinocytes, and that's a special type of secretion. We call it cytokine secretion, where you pass cells from one cell to another cell specifically, and that's what happens in melanin granules. They're produced by one cell, but they actually are housed in the keratinocytes. So here you can see the clear cells of melanocytes located in the basal, and then they pass their granules on to the keratinocytes. So these are melanocytes, keratinocytes here. You note that the melanin is really above the nucleus, it's capping, as we call it. So here we can see skin, stratum corneum, stratum granulosum, stratum spinosum, stratum basal, and here you can see the melanin granules, which are above the nucleus. So you're trying to protect the nucleus in the basal, because indeed that is a source of the cells that are mitotically active, and then these cells differentiate from that mitotic activity. So here we can see the melanin capping on those cells to protect. Here's the sun, NASA's sun, and so UV detection will be coming through here, and you have these granules right above the nucleus that you're trying to protect. You don't care about these nuclei, they're differentiating anyhow, but you do care about the nuclei on the base. Here we see electron microscopic level. You can see one of these keratinocytes with the melanin granules capping above the nucleus. Also, here we see a melanocyte with its dendritic processes projected. You can see them projecting it through there, which is a source of the granules that are located in the keratinocytes. Now, melanin granules accumulation over the nuclei of mitotically active cells of the stratum basal is important to protect the DNA against the UV light, as we mentioned before. Also, melanocytes are responsive to melanocyte-stimulating hormone, which comes from the pars intermedia. And in fact, melanocytes are susceptible to different things, hormonal physical things. Sun tanning, for example, will stimulate melanocytes to secrete more melanin granules. Also, sex differences. Testosterone influences melanocytes, and indeed it's more males are darker than females. Pregnancy will cause a change in pigmentation of women. So there are different things that can affect the melanocyte action. Also, melanocytes vary in the people. The density of melanocytes is similar with the different races or different colorations of skin. What is different is the size of the melanin granules. Here you see in a Caucasian where you have fairly small melanin granules, and they're actually contained in a membrane. as you see there, which pass from the melanocyte to the keratinocyte and through there. In a black-skinned individual, what happens is the melanin granules are very large, and they stay with their own membrane. They don't coalesce in a group like you see in a Caucasian. And here you can see this is Caucasian, this is Asian, and this is an African descent person. Now also what happens is sometimes freckles happen where you get the melanin granules are distributed in patches instead of being distributed uniformly in the skin. They're distributed in patches, and that's what creates freckles. There are diseases associated with melanocyte. Of course, albinoism is a failure to produce melanin or a little bit of melanin, and here we see albino rat. The pink that we see there is actually from the blood, from the red blood cells that we see. Otherwise, there's no pigmentation in the hairs, and the skin you can see is albino. Also, cancer, malignant melanomas are not a good thing to have. And Addison disease, the pigmentation is positive, kind of like freckles in the skin, due to adrenocortical insufficiency in the Addison disease. And we'll talk about clinical correlations later on about other ones. But Langerhans cells is another cell type. It's from the bone marrow, its origin, where most immune type cells originate, and they're located in the stratum spinosum. So a clear cell in the stratum spinosum, they don't make desmosomal connections, and they appear clear cell would be likely going to be a Langerhans cell that we see. Clear, no desmosomal connections, but they are dendritic in process because they have an immunologic role to play. So one characteristic of these is that the granules inside there are racket shaped, look like a tennis racket. Characteristic of these granules. They do have immunologic roles play, so they are able to receive antigens that may be coming through the epidermis, contact allergic responses, and other cell-mediated responses in skin is the function of these Langerhans cells. Now there is an epidermal-dermal interface, as we talked about before, the thin skin versus a thick skin. This thick skin is usually more rough, more smooth with the thin skin. And you can see that the thick skin has an elaborate, the dermal papillae are much more elaborate than what we see, bigger dermal papillae over there in thin skin. And this interface between the dermis and the epidermis is to prevent, whenever this is resistant mechanical stress, it doesn't separate the epidermis from the dermis. So if we pull the epidermis off, you will see little holes in through there. This is where the dermal papillae projected into those holes. Here we can see melanocytes, the clear cells in through here, and melanin granules that are produced here. But also we can see the dermal papillae projecting up through there as well. Here we see the melanocytes. If we pull the epidermis off and look on the side, this would be where the dermal papillae was in through there, and then this would be the epidermal pegs that you see there, and here we can see the melanocytes on the bottom of this surface. This would be hairs of a face where you have a beard. This is a finger pad, and this is the belly, I think. So there are differences in their interfaces with the different regions. Finger pad, you have to have more elaborate connections between the epidermis and the dermis than you would, say, in the cheek. We can see more. This is a finger pad. There's a finger you can see where you have the epidermis-dermis interaction in through there, making these dermal papillae that project up. Remember that the stratum basale is the source of the cells. So there's more of these mitotic active cells here than there would be here. And as a consequence, there's more other products pile up here and pile up there. So every time you have an indentation, you're going to have a pileup of cells at the surface. And that's what makes our fingerprints. Even the soles of the feet have this type piling up of things. And that makes the fingerprints. That's what we actually see in the fingerprints. And there are different types of swirls that would be because it's a different type of dermal epidermal interaction that you could see. Here we see a presenting corpuscle, but here in thick skin you can still see the piling up of the cells in the stratum corneum. Also, we see the presenting corpuscles. The presenting corpuscle look like onions, as we see, and the presenting corpuscle is a mechanical receptor too that detects vibration and pressure. So pressure, as opposed to the Meissner's plexus, was light touch. This is more pressure and vibration, and it's usually it's deeper in the dermis. Also, we see sweat glands there. In thin skin, we can see hair follicles. Hair follicles project up through there. This is a smooth muscle here, a rector pili muscle that pulls the hair up. If you've ever seen a cat that's upset, the hair is sticking up, their rector pili muscle is pulling the hair up. Also, the hair is pulled up when you're cold, and as a consequence, the surface of the epidermis makes a little indentation. We call that chill bumps or cold bumps, which is really the effort of the skin to pull the hair up to warm. So also we see a sebaceous gland. Sebaceous gland discharges contents in the hair follicle. So here we see the hair follicle. This is actually the hair itself, which will be projecting through there, and it's just extension of the epidermis down deep within the dermis, and the hair follicle is produced. Here's a rector pili muscle in the slide 31. The follicle itself, sebaceous glands, which discharge your contents. Now, if you have straight hair, you will have straight follicles. However, if you have curly hair, you will have curly follicles. So curly follicles produce curly hair. And here we can see this. This is a scalp of a human showing you the curved follicles, which gives rise to curly hair. And here we can see the sebaceous glands actually dumping into the hair shaft here. So this is actually the hair itself that we see there. And it's in this region that we have the melanocytes that give the hair its color. Again, we can see the hair follicle itself. The growth occurs from down here. Hair projects in through there. Sebaceous gland, as we can see. And the mode of secretion of sebaceous gland is holocrine secretion, where the contents is discharged when a cell bursts. When a cell bursts, the whole cell is the secretion itself. Also, that brings us to the eccrine sweat gland. Here we see the sweat glands. Here's the sweat gland duct, which is more eosinophilic than is the sweat glands itself, and it'll be discharging on the surface. Sebaceous gland discharges in the hair follicle, but the eccrine sweat gland discharges on the surface of the skin, as we'll see a little later on. So here's the eccrine sweat gland. There's a secretion portion, and then there is the duct portion, which discharges on the surface of the skin. But this is more what we see. So this is typical of what we see. There's the duct itself. There's the secretions of the eccrine sweat gland. Sebaceous gland, as we see in 66, and the hair follicle itself, and the eccrine sweat glands. Now, in this slide 66, we have two types. We have skin, which is stratified squamous epithelium in the anal wall. And so that's what we see, stratified squamous epithelium. But in contrast, we also have a mucosa, simple columnar epithelium of the rectum with goblet cells. There's even goblet cells that we can see there. This is a mucosa that we see, the epithelium of the mucosa. Lamina propria would be in here, as opposed to here would be dermis. So we have epithelium on both, stratified squamous, simple columnar, dermis, lamina propria in through there. So here's the sebaceous glands that we can see, and this is the apical sweat glands. Apical sweat glands are different from the American eccrine sweat glands in that they thought that the motor secretion was apical. Now we know it's not, but they still keep that name. And these, so that brings us to the types of secretion. American secretion is simply exocytosis. That's how proteins get out. Apical secretion is where some of the plasma membrane itself goes out with a secretion. And if we look at milk producing cells, they have both of those. They have American, which is how you get the milk protein, casein, is discharged by American, but also the milk fat droplets, as we see here, are discharged with some of the plasma membrane around it to make the fat water soluble, because fat is not water soluble. You wouldn't be able to get the fat out of the milk if it wasn't for the fact that you have the plasma membrane wrapped around the fat droplet to keep it solubilized in the milk. The other type of secretion is Holocrine secretion, and that's what happens with the sebaceous gland, where the secretion is the cell itself. When it pops, all its discharge is cell contents are indeed the secretions itself. Now, the eccrine sweat gland for cooling discharges on the cell surface. That's your common sweat gland. But the apical sweat gland discharges in the hair follicle itself, because this is used for like marking of animals, to mark territory, and it'd be easy to mark if you had a brush. And of course, the hairs would be the brush that the animals would use to mark. So apical sweat glands usually in the axillary regions of the humans, but in animals it's used to mark the territory, and it discharges in the hair follicle, as opposed to this guy, the function is cooling, and it needs to be on the surface of the skin. And you can see where a typical common sweat gland discharges different places along the fingerprints. So you can see why you would leave fingerprints, because it's discharging right on these grooves. And the surface of these grooves, of course, is the abundance of cells in the epidermal pegs that project down into the dermis and pile up on the surface. And these piles that we see there with the sweat discharge on it leaves our fingerprints. And so here we see the duct and the secretory portion of the sweat gland. And here we see the myoepithelium. The myoepithelium, we can see it's a kind of pink that goes around there. The myoepithelium squeezes the secretion portion. You don't have it on the duct, you just have it in the secretion to squeeze the secretions out, and you see it's kind of pink. Myoepithelium is eosinophilic because of the smooth muscle contraction proteins. So it's proteins that is associated. It's not smooth muscle. It's actually epithelium. However, it does have actin and myosin proteins in there, and that's what makes it eosinophilic. And whenever it contracts, it discharges the sweat content. And here we can see the myoepithelium. These are epithelial cells, now not smooth muscle. And you can see how it forms like a little mesh around it and squeezes the secretions out. So in functional diversity, remember there's a lot of things. It protects against injury, against UV light, mechanical stress, desiccation. Those are some of the things that it did. The melanin granules is one of those that's associated with, it's produced by the melanocyte, carried on to the keratinocyte, and here we see some melanin granules in there. Also, there's a membrane coating granule that is discharged at the stratum granulosum, and the membrane coating granule coats the membranes for the stratum corneum. Then we have the keratin-hyaline granules, which are the intermediate filaments that are the contents of the cells of the stratum corneum. So here we can see the membrane coating granules, also called lamellar granules, because they have different layers that whenever it discharges contents, it seals between these cells of the stratum corneum and stratum granulosum, which causes prevention of dehydration to occur. So the keratin hyaline granules is really chemical nature is not known. They're rich in histidines and is very insoluble. Here we see the keratin hyaline granules here for the stratum granulosum in through there, and that's the only content that we have in these. So in terms of regeneration of the epidermis, if you have a source of cells, you can regenerate the epidermis on the surface. In fact, the degree of burn depends upon how deep the burn is. If it's below the hair follicles, you have to have a skin graft. But if it's above the hair follicles or above the glands, also the sweat glands would be a source of epithelium to repopulate the surface as well. But if a burn is below that, you will not have enough cells to repopulate the area. So in terms of our clinical correlations of patients with albinoism, would be more susceptible to basal and squamous cell carcinoma because they don't have the pigmentation above the mitotic active cells, and they'll be susceptible to DNA alteration due to UV exposure. Thank you.